Hello, everybody. I'm Mr. Davenport, and welcome to our week's lesson for Introduction to Biotechnology. This week, we are talking about notable people in the history of biotechnology. And this is going to seem kind of like a repeat of how we were discussing the notable applications because there are going to be some details that are similar. But also, I'm going to provide kind of a biographical focus on some of these important figures. The website that we are going to be pulling this information from is from one that I've created, and I'm going to be posting this website in the canvas so that you guys will be able to access the exact same information. Also on the website, there are translations for those of you who need Spanish translations of this information for further understanding. So, let's get started. Our first notable person in the history of biotechnology is Hippocrates. Hippocrates was born in ancient Greece at 460 BC. And his father was a doctor. And as a young man, Hippocrates traveled the world and became, at least the world that they knew it at the time, and basically learned a lot about medicine from his father and other physicians. Hippocrates studied the human body and made observations of his work, and eventually he developed his own theories about how illnesses impacted people and rejected the current belief that illnesses were that they were curses or that they were caused by evil spirits or because someone made the local river god angry. He rejected those ideas and believed that illness was something that came from the outside and attacked people. Okay, it came from the outside and infected people, which was kind of a modern idea at the time. Hippocrates founded the first school of medicine on the island of Kos and began teaching his ideas. And he taught that the body should be treated as a whole. It's called a holistic approach. And he believed that treating illness with rest, a good diet, and fresh air, and being clean was the way for individuals to stay healthy. And Hippocrates is known as the father of medicine. He's credited with developing the ideas and principles of the Hippocratic Oath that's still taken by physicians today before they begin their medical practice. And Hippocrates died in 377 BC. Our next notable person is Louis Pasteur. Louis Pasteur was born in a small town in France in 1822. His father was a leather tanner, and Pasteur was a bright student, and in 1847 he had earned his doctorate in physics and chemistry. And he's credited and continued his research for many years and is credited with a huge number of discoveries. For example, Pasteur founded the science of microbiology, basically meaning he was the first person to study microorganisms in depth and how to grow them, which eventually led to how do we stop them from growing? How do we fight bacteria? He also proved the germ theory of disease and invented the process of pasteurization, which is if you drink milk or eat cheese or eat ice cream, you should thank him for, because pasteurization is the process of killing a lot of the natural bacteria that can spoil milk and by heating it up and then keeping it cool to keep bacteria from growing. He also discovered that weakened forms of a microbe can be given to a person who, and then an individual can develop their own immunity to that disease. Because he discovered that weakened form process of exposure, he developed vaccines for several diseases, including rabies and anthrax. And Louis Pasteur is responsible also for influencing changes in hospital practices to minimize the spread of microorganisms and infections. Pasteur died in 1895 at the age of 73. Our next notable person in biotechnology is Gregory Mendel. So, Gregory Mendel was an Austrian monk born in 1822 in Austria. And he was born to very poor parents. At the age of 21, he entered a monastery to study philosophy, theology, and natural science. And his study of natural science led him to want to experiment with flowers in the monastery's garden to try to come up with different color variations through selective breeding. And it was in these experiments that Mendel started playing around with what was called hybridization, creating crossbreeds. And between 1856 and 1863, he cultivated and tested almost 28,000 individual plants. He did much of his research with pea plants because they were easy to obtain and easy to grow. Mendel's work was not well understood or appreciated in his lifetime. It wasn't until the 1900s that three scientists from three different countries in Europe started doing similar experiments and came across Mendel's research. And basically, through his remarkable research on plant hybrids, Mendel established that there does exist in living things dominant traits and recessive traits. And this is what we call Mendelian law. Because of this, he is often called the father of genetics because it was that basic idea of dominant traits versus recessive traits that started to guide the practice of genetics. Now, we know now that genetics is a little more complicated, but the basics of Mendelian law still exist. There are some traits that we consider as either dominant, you express it and have that trait, or it's a recessive trait. You don't have it, and it's not, or that trait is not expressed in a certain way. We'll explain a little bit about that a little bit later. Gregory Mendel died in Austria in 1884. Our next notable person in biotechnology, Rosalind Franklin. So Rosalind Franklin was born in London in 1920, the daughter of well-to-do Jewish parents. When she was 15 years old, she announced to her family that she wanted to be a scientist, and in 1938, she entered Cambridge University, and where she earned a PhD, and at the age of 26, she began working on X-ray diffraction, which is a process of using X-rays like we would take with broken bones and to see inside the human body. To create images of crystallized frozen solids. And this was used to kind of map how different proteins looked to get kind of their 3D image because they couldn't see them under a microscope. They shot X-rays literally at these crystallized solids, and they could apply an algorithm to get what was called a diffraction pattern so that they could then use math and a massive algorithm to determine the shape of that protein. Really complicated stuff, highly advanced science, especially for that time. In 1950, Rosalind Franklin was invited to work at King's College in London and to join a team of scientists who were studying living cells, and she was assigned to work on this DNA project with a gentleman known as Morris Wilkins. And Morris and Rosalind did not get along. Rosalind Franklin knew what she wanted to accomplish and went about her business, and Morris Wilkins, who was less trained than Rosalind Franklin, and a little jealous, and a also big personality clash led to them just not being able to get along. And so this made both of them unhappy to the point where after 2 years at King's College, Franklin's research advances in X-ray diffraction techniques and her techniques of studying DNA and being able to determine there were two types of DNA, A DNA and B DNA, which led to greater, better pictures. All of these led to crucial clues and keys to how DNA's 3D structure existed. Problem is Morris Wilkins somehow, I'm not going to say he stole it because there was a whole bunch of confusion in the story, but Morris Wilkins somehow got Franklin's data and shared it with an opposing team at Cambridge University, which was James Watson and Francis Crick, who are now credited with the discovery of DNA structure. But Crick and Watson would not have been able to achieve their model and their publishing if it were not for Rosalind Franklin's background work. She proved the entire structure of DNA without help and without Watson and Crick. Watson and Crick put the information together based on info that they had been discovering independently, but it was essentially Rosalind's work that proved DNA's three-dimensional structure. Unfortunately, Rosalind Franklin was not recognized for this achievement, and she died of ovarian cancer in 1958 at the age of 37. That leads us to our next individual, James Watson. So in 1963, along with his colleagues Morris Wilkins and Francis Crick, James Watson was awarded the Nobel Prize for his work in the discovery of DNA structure in 1953. James Watson was only 25 at the time of the discovery, and he was born in Chicago in April 1928 and moved to work in England at the Cavendish Laboratory at the University of Cambridge. Watson had a master's degree and a doctorate in zoology, he became interested in genetics. Watson truly believed that studying the gene, which would eventually lead to the study of DNA, was how he would discover the three-dimensional structure of DNA, which was the problem they were trying to solve at the time. So from 1956 to 1978, Watson was on the faculty of Harvard University in the biology department, promoting research in molecular biology. From 1968, he served as the director of the Cold Spring Harbor Laboratory, greatly expanding the level of funding and research, which was a DNA genetics research organization. The Cold Spring Harbor Laboratory, he shifted his research to the emphasis on the study of cancer, along with making it a world-leading research center in molecular biology. Between 1988 and 1992, Watson was associated with the National Institutes of Health, or the NIH, in helping to establish the Human Genome Project. In 1994, James Watson served as the president of the Cold Spring Harbor Laboratory and served there for 10 years, and then he was appointed as chancellor, serving until he resigned in 2007. Problem is, James Watson, despite all of his accomplishments, at his core, he was kind of a jerk. It was his idea to basically take and steal Rosalind Franklin's information that led to he and his partner, Francis Crick's, publishing of their work, which basically gave them all the credit and Morris Wilkins an assisting credit for the discovery of the structure of DNA. So Watson, at his core, not a great guy, honestly. Did a lot of great work, but not a wonderful human being. On the other side is Francis Crick, who expressed great remorse for basically how Rosalind Franklin was treated by both Morris Wilkins, and James Watson. And just quick look at Francis Crick. Crick was in 1963 also recognized for the discovery of DNA that they discovered and published in 1953. Again, a partner in crime of James Watson and Morris Wilkins. But basically, Francis Crick over time realized how wrong he and Watson and Wilkins, and even Wilkins started to come around later in life about how wrongly they had treated Rosalind Franklin. And it really upset him that it was ultimately they got recognition for something that they knew they did not only had a moderate part in. I don't want to take away their accomplishments as scientists because they are very accomplished, well deserving of their credit because they did put in the work. But they could have no way have proven the theory of their three-dimensional structure of DNA, which we still use today, without Rosalind Franklin's research. So Crick was a biophysicist and was born in Northampton, England in 1916. He helped develop radar and magnetic mines during World War II. And after the war, he started researching DNA at the University of Cambridge, which is where he met James Watson. Shared the Nobel Prize for Physiology in Medicine with Watson and continued his conducting research until his passing in 2004. All right, our next notable person, Herbert Boyer. So we know that Herbert Boyer was a part of the team that discovered recombinant DNA, but there is more to one Herbert Boyer than that. Give me a moment. I accidentally clicked out. I need to zoom in on some stuff here for you guys because I accidentally cut it out. Don't know how I managed to do that. So, guys, Herbert Boyer was born in Derry, Pennsylvania, and in July 10th, 1936. He played lineman on his varsity football team, and he was always wanted to be a doctor. After high school, he went to St. Vincent's College and started with the pre-med program. It didn't take long for Boyer to realize that being a medical doctor was not what he wanted to do after all. So by the time he graduated college in 1958 with a Bachelor's of Science in Biology and Chemistry, Boyer had decided to become a researcher. And he did graduate school work at the University of Pennsylvania and then postgraduate work, doctorate work at Yale. In 1966, Boyer accepted an assistant professorship at the University of California in San Francisco. And he became interested in E. coli bacteria, especially in restriction enzymes that could be isolated from the E. coli bacteria. These restriction enzymes basically allowed for the cutting of DNA in certain sections. And so at a conference in Hawaii in the 1970s, Boyer met Stanley Cohen, who was working on plasmids, which are these little rings of DNA inside of bacteria. And the two began a collaboration on their work, which led to the creation of the first recombinant DNA. In 1975, Boyer met an individual named Robert Swanson, who worked for a venture capital company. Swanson believed in the burgeoning biotech industry, and in 1976, Genentech was born, which was the brainchild of Robert Swanson and Herbert Boyer. Genentech cloned and later developed a method for synthesizing human insulin using recombinant DNA technology. Genentech continues to be one of the biggest biotech companies in the world. In addition to founding the company and creating the biotech industry, Boyer, along with Cohen, patented the recombinant DNA technique. This is one of the first biopatents granted.
Will all math teachers please head to Ms. Melissa?
In addition